Now let us do an experiment for photosynthesis. Aim is to prove that chlorophyll is essential for photosynthesis. Apparatus required are a potted plant, a sheet of paper, pen or pencil, water beaker, alcohol beaker, water bath, pipe Bunsen burner, iodine solution beaker. Procedure: take a potted plant with variegated leaves, for example, money plant or crotons. Keep the plant in a dark room for 3 days, so that all the starch gets used up. Now keep the plant in sunlight for about 6 hours. Pluck a leaf from the plant. Mark the green areas in it, and trace them on a sheet of paper. Dip the leaf in boiling water for a few minutes. This process destroys all the enzymes in the leaf and stops any further chemical changes taking place. After this, take out the leaf from boiling water, and then immerse it in a beaker containing alcohol. Now place the beaker carefully in the water bath and heat till the alcohol begins to boil. We see that leaf becomes colorless, as it loses its chlorophyll. Now dip the leaf in a dilute solution of iodine for a few minutes. Take out the leaf and rinse off with the iodine solution. The green portion of leaf turns blue-black, indicating the presence of starch. The non-green portion of leaf turns brown, indicating that starch has not been formed. Conclusion: as only green portion of leaf turn to blue-black, this proves that photosynthesis cannot take place without chlorophyll. Let us do another experiment for photosynthesis. Aim is to prove that carbon dioxide is essential for photosynthesis. Apparatus required are 2 healthy potted plants, watch glass containing potassium hydroxide, bell jars, vaseline, iodine solution beaker. Let us do the experiment. Procedure: take 2 healthy potted plants which are nearly same size. Keep the potted plants in a dark room for 3 days. Now place a watch glass containing potassium hydroxide beside one of the potted plant. Potassium hydroxide is used to absorb carbon dioxide. Next, cover both plants with separate bell jars as shown in the diagram. Use vaseline to seal the bottom of the jars to the glass plates, so that the setup is airtight. Keep the plants in sunlight for about 2 hours. Pluck a leaf from each plant, and check for the presence of starch using iodine test. That is, dip the leaf in a dilute solution of iodine for a few minutes. After that, take out the leaf and wipe it off with water. The leaf which was exposed to iodine test turned to blue-black. This shows starch is formed by the process of photosynthesis, using carbon dioxide in the bell jar. The leaf from the plant, that is potassium hydroxide in the bell jar, does not become blue-black. This means photosynthesis did not take place, in the absence of carbon dioxide in the bell jar. Conclusion: this experiment proves that photosynthesis cannot take place without carbon dioxide. So far, we have discussed about how autotrophs meet their energy requirements. But they also need other raw materials for building their body, that is, water. Water is another important raw material required for photosynthesis. Terrestrial plants take up water from the soil through their roots. Carbohydrates formation is not possible without water. Apart from water, plants also absorb nutrients like nitrogen, phosphorus, iron and magnesium are taken up from the soil. Nitrogen is an essential element used in the synthesis of proteins and other compounds. This is taken up in the form of inorganic nitrates or nitrates. A bacterium converts atmospheric nitrogen during nitrogen fixation, which are neutralized by the plants in the form of ammonia, that is, NH3. Till now we discussed about the nutrition in autotrophs.